So, good afternoon and welcome to this hybrid seminar for staff here at the LMB and for our online audience as part of the Biomedical Campus virtual tours. So, before we begin, just some technical details. At the end of the talk, there'll be the opportunity to ask questions, and I'd really encourage you to do so. If you are participating with the seminar online, please write your questions in the Q&A, and then I can read them out at the end so the speaker can answer them, and also so that the in-house audience can know what you've asked. So before we start, just a few words to introduce the LMB to our online audience. So the LMB MRC Laboratory of Molecular Biology, also known as the LMB, is a world-leading research institute dedicated to understanding complex biological problems at all levels, from atoms and molecules to cells and organisms. We aim to study difficult long-term problems and in doing so contribute knowledge needed to solve key human health issues. So over the years, LMB scientists have made many contributions to medicine, both in terms of new understanding and in the development of new technologies. And today I'm delighted to welcome Albert Cardona from the Neurobiology Division to give our talk. So Albert completed his PhD in developmental biology at the University of Barcelona in 2005. He then spent a short time working as a software engineer at the Institute of Neuroinformatics in Zurich before completing a postdoc in the neurobiology of the fly at UCLA. In 2008, Albert was awarded a group leader position at the Institute of Neuroinformatics in Zurich, where he developed software to aid analysis of neural circuits and to enable and support his research on fly connectomics. In 2012, he moved to Howard Hughes Medical Institute at Janelia Research Campus, where he organized dozens of laboratories worldwide to jointly map the wiring diagram of the larval fly nervous system. And in 2019, Albert moved to Cambridge, was awarded a fellowship at Pembroke College, and became a program leader here at the LMB to conduct research on neural circuits within the newly formed Molecular Connectomics Initiative at the MRC. So today he will tell us about the brain, what for.
Microphone's working? Yes. Great. Hi, everyone. My name is Albert Cardona. Thank you so much for the introduction. Thanks for your interest in this topic. It is not the easiest topic to speak about, as we all have brains, and we all very much care about them. So, when it comes to the brain, there's this perspective from philosophy or so on to overthink it, to really consider the brain fundamental, necessary, essential, you cannot live without it kind of thing. But the typical joke is there are many occasions in which having a brain is not necessary, can even be detrimental. If you are mating and you're a mantis, you're probably going to lose your head. But for a good cause, nutrition towards the next generation. So let's explore now, in a simpler organism, what is the brain for? But before that, realize across the tree of life, the many animals and life forms, there are many occasions where really the brain is not, it just gets underway. So here we have an abyssal angler fish. And what happens here is the females are large, the males are small, and the ocean is vast. And therefore, when they disperse, finding a female is not easy. When you find one, you want to keep it. How do you do that? Well, the male latches onto the female with its useless teeth. And then, essentially, its body kind of dissolves, and it becomes an appendage, literally a permanent attachment, with a shared circulatory system, the blood, with the female. And then, only then, the male's testes start developing, and it becomes essentially, sperm on demand. When the hormones come in, it will just release it. I mean, why do you need a brain for that? You needed it briefly to find a female, essentially. And that was that, right? So now, let's go on to an animal where we can study what is the contribution of the brain to the survival and life and behavior of the animal. And so in the lab, among other animals, we work in Drosophila. And in particular, we don't work in big Drosophila larvae. We work in smaller and small, tiny little Drosophila larvae. Why do we work with animals so small? It's because they are tractable. Our techniques enable us to look at the brain, to look at the neurons, to look at the synapses that relate these neurons to each other, and actually make sense of them. And so if we look at the head of this animal, it's essentially just the anterior end. It's not necessarily very different from the rest of the body. What we see here are these repeated groups of sensory neurons on the skin of the animal, which have been labeled, and here they are black. And then each one of these projects to the central nervous system, where we have the homologous of a spinal cord, like we have in our own bodies, as well as the brain, this tiny little agglomeration of neurons right there. There, the scale bar is only 0.5 millimeter. And so, what does this animal do? Well, it has to crawl around, explore the environment, shift its head left and right, find places that are not too hot, they're not too cold, find places that there's not too much salt, there's not too much sugar, right? It really wants to live comfortably and nicely. It needs to also find food, and it needs to avoid predation. That's pretty much it. Not a very complicated life. And yet it has a brain with 3,000 neurons. Why would you carry the cost, the metabolic cost of maintaining, building, and so on, this huge amount of neurons if it was worthless, right? So let's have a look here. I said every segment has this, every block of the body, every segment has this set of sensory neurons. In addition, every segment has a bunch of muscles, and they look very similar from one segment to the next. We are also segmented. You can find the muscles if you count your abdominals, if you can see them, if there are enough exercise. And so, being segmented is something that most animals are. There's nothing there to it. But the point is that these segments have muscles, and these muscles are connected in such a way that enable the animal to contract these segments. And that's how it can move. So let's have a look at that. The brain is just right here, this tiny little blob at the anterior end of the central nervous system. And so here's a video showing an animal moving from left to right. These vertical green bands are labeled cells in each of the segments of this body. And unfortunately, this video didn't play again, which it should have. And if it, well, I can just go back and play it again. Can I? No. Uh-oh. There we go. Right, as it plays again, you can see that these bands, which mark each body segment, are moving towards each other. As they move towards each other, remember, you have some of these segments more in the so-called the thorax, like this part, right? And then in the abdomen, which will be the bottom part of the body. And so now, if we look over time, what happens to these segments is we can see the contractions. This is what this is measuring. And so the contractions in the posterior, towards the end of the animal, towards the tail, happen first, and then in the more anterior bits until the very front bits. And so if you follow this dashed line, you see that there's a contraction moving forward. This is a wave. It's called a peristaltic wave. And that's how this animal moves forward, like an inchworm, right? And now, now that you understand how this moves, we can now make a little experiment. Suppose we get rid of the brain. How do we get rid of the brain? There are many ways, but that's why we work in this animal, because we can do that and the animal will still be fine, right? Drosophila, for 100 and something years, has been an outstanding experimental model system precisely because we can do very fine-grained genetics on it. How do the genetics work? Is you have a genetic construct in one fly, so the so-called GAL4 system. This was stolen from yeast, from a little yeast, the Saccharomyces cerevisiae that you use to bake bread or to make beer. We stole a cassette of genes from there and put them into the fly. And then what this cassette does, this protein right here, is basically drive gene expression. It activates a gene. But to do that, it needs to bind somewhere on the genome so that then it will tell, the DNA polymerase, like, can you now copy this gene and start the process of expressing this gene? So it needs something called the upstream activation sequence, which is nothing else than a chunk of DNA with a particular sequence that this protein will recognize. And so if you now have one animal, in this case, say, the female, that is expressing this protein in a particular set of tissues, such as the brain, and you have now another animal that has that particular activation sequence, that which gene will activate? In this case, the green fluorescent protein, which we stole from a jellyfish, right? If you study jellyfish, you will end up transforming neuroscience. Who would have thought, right? And so now you put the two together, and the F, meaning the filial, so the children generation, will hopefully have both. And when it has both, now we look at the tissues. Here's the little maggot. Here's the brain, fully green, expressing the jellyfish fluorescent protein. If we can express the fluorescent protein, we can express other things, many other things. And here's where things become interesting. So what can we express? Well, to switch off the brain, we need to modify the activity of the neurons in the brain, for instance. We could kill them, but that would be kind of dramatic. Why don't we just inactivate them? We can do that by targeting, in this case, we use the same protein, the same construct that will target expression in the brain, but on top of it, we say, well, there was another gene in that yeast cassette of genes that we stole, right, to do genetics in the fly, which what it does is it stops that protein from activating gene expression. And so this protein is called GAL80, that's the suppressor. And now we're saying, okay, we're going to use another promoter in the genome that is triggered only in the equivalent of the spinal cord. And so now, instead of expressing in the whole brain, we're going to say, we don't want expression in the spinal cord, we only want it in the brain. And that works. It's called T-shirt GAL80, TSH. Why is it called T-shirt? I'm not sure. But Drosophilists really like to name things in a memorable way. And so it's not perfect. There are a few neurons that escape that suppression, but by and large, the entire spinal cord is free, and it can work as it should. And so we see it here in fluorescence, here in a different way. It doesn't matter. It's just to show the brain lobes will now express what we want, and the nerve cord will not. So what do we want to express in the brain to stop it from working? I'll show that in a moment. Basically, if we look at the larva, this tiny little animal, as simple as it is, it will turn. How does it turn? It does a hemilateral contraction. Can I imitate that? Just like that, right? And then if we now look at the angle of these turns, are they big, are they small, and the frequency of these turns, in three separate conditions. What are these conditions? The first control is we're going to express a construct that is called Shibiri temperature sensitive. It doesn't matter. What it will do is it will prevent the neurons from firing onto the next, from passing the nervous impulse onto the next neurons. And therefore, the circuits will be stalled because whatever inputs they receive, nothing will go through, right? But we only put that protein. We don't tell the animal where to express it. So it is there in the genome, silent. If there's any leakage, it would alter our experiment. Therefore, that's why we put it as a control. And what we see is that these animals are turning in this little diagram. These are angles, 180, and so on. So these animals are turning 45 degrees one way, minus 45, 45 degrees the other way. And the magnitude of this little triangle is, how often do they do these turns? And what we see is that this is not different than wild type. Here we have another control. This control is, now we don't have that effector protein that would suppress synaptic transmission. Instead, we have the actual other gene that would control where the inactivation will be. But it controls nothing because there's nothing to express. That's another control because we're basically trying to see what happens when you have only half of the system, but not the complete genetic system. And again, nothing happens. There's a perfectly fine amount of turning in minus 45 plus 45 degrees as the animals explore the substrate where they live. And so now we're going to inactivate the brain by putting both together. This brain lobes sort of genetic construct that controls expression only in the brain, right? Together with the temperature-sensitive Shibiri, which will suppress the release, the synaptic release. It will suppress the transmission of the nervous impulse. And as you do that, what happens? Nothing. You still have, animals starting 45 degrees one way, 45 degrees the other one, exploring the environment as if nothing has happened. It's like, wait, what? So that movie that I showed you before of the animal happily exploring the environment, you can totally do that without the brain. It's like, okay, why does it need a 3,000 neuron brain then? So nothing happens. The animal with the brain switched off explores the environment in a way that is indistinguishable from controls. So now let's give it a test where we are hoping, we're rooting for the brain. Like, you have to do something, right? What do you do? So what does the test consist in? Basically, what we have here now is a different kind of arena where we put the animal in a place where there is a little odoron that it likes, say, smell of chocolate, something that you really want to go towards. And the colors simply indicate the concentration of that odor. So the periphery has few in blue, and as you go towards the center, there's more and more concentration of that odoron. And so now we take our two controls. Again, one is the construct that would restrict expression in the brain lobes, but there's no effector, so the neural activity will not be modified in any way. And this animal, wherever it starts, it doesn't matter, it will climb up gradient until finding the source of the odor and kind of hang around there in this high gradient place, right? The same is true if we now put a different kind of effector. It's not that temperature-sensitive CBD that prevents synaptic release, but actually is a way in which through light we can express, we can switch on a chloride channel that will hyperpolarize these neurons. What this means is these neurons will be in a state that they cannot emit action potentials, they cannot communicate to other cells. But we can do that with high temporal resolution because we can switch it on and switch it off with light. But again, we put that effector, but we don't tell it where to express it or how to express it, so we're just testing for leakage. Is it expressed anyway? Right? And it's not. This animal is perfectly fine. It can just crawl, again, up gradient, find the source of the odor, and hang around there in frustration because it's just an odor, there's no food. But now we put both together. We inactivate the brain. What happens now? No matter where the animal starts, it will walk around and completely ignore the gradient. So yes, it will crawl around in a way that is indistinguishable from, say, a wild type, but the moment there is a task where you have a direction, where you have something to do, now it cannot do it. So this is so interesting that a group in Edinburgh, for Barbara Webb's group, Jane Lovelace, a student in her lab, decided to model this animal without the brain. And said, how is it possible that just the spinal cord, right, just the biomechanics of the body, just the basic things that are not the brain, are able to produce behavior that is so indistinguishable from a complete animal with a functioning brain? And what they did is the typical approach of modelers. What is the simplest thing that could possibly work? And they took every segment of this animal and simulated it with a little spring. And then they said, we're going to connect these springs. Well, first we're going to contract these springs with a motor neuron, like a neuron that is able to, say, contract muscles, right? And this contraction will be sensed by a sensor neuron, a stretch receptor. So when you stretch your arm, you know you're stretching your arm because there's a stretch receptor that says, this muscle is being extended now, right? And so here's in green the stretch receptor, in red is the motor neuron. And so now, if the motor neuron is driving the spring will relax, and then the stretch receptor will sense that, and it will maybe suppress the neuron, or the other way around. The stretch receptors normally are always active, and when the body segment is contracted, then they stop being active, so then the motor neuron stops triggering that, and it relaxes again, and starts again. It's a kind of dynamical system. And then they added one single circuit element, which is, can we add an interneuron that will take the input from the stretch receptors of one segment here and suppress the motor neuron of the adjacent segment. And that's it. There's nothing else to this model. It's as simple as it gets. So what do they get with that is the following. As the animal crawls forward, right? What you get here, each of one of these dots represents a segment of the animal. And you can see that they become closer to each other as the wave of contraction goes through, and further apart as the wave goes away. And that's how it inches forward. It's essentially a bunch of dynamics that can be simulated very simply. And what's more interesting is if you now simulate hundreds of these animals, each one of these colored little stripes is one of these simulated animals in the computer. They're crawling around. They're expanding, they're exploring the environment in a pretty much completely Gaussian way, as in every direction possible. All of these are simulated larvae, simulated tiny little things with springs, a motor neuron, a stretch receptor, and inhibitory cells to the adjacent segment. There's nothing else to it. There's no brain here. There's like the modicum spinal cord, right, which is like the inhibitory cell that goes to the adjacent segment. And yet, as a Drosophilus, I look at this and I cannot distinguish these animals from wild-type animals, from real-life animals, right? They crawl just fine. So, okay, before I get to that, bottom line, the body does a lot more than you would ever give it credit for. As you walk around, you're not thinking where you're going to put your toe, your knee, your whatever, right? The spinal cord is doing that. And beyond the spinal cord, the biomechanics of your muscles, the elastic characteristics, the rigidity of the bones, all of that is doing a huge amount of work that we take for granted. And all the brain has to do is give vague directions. Stop now, walk forward and go backwards, modulate the speed. It doesn't really need to specify everything. And so how does the brain do that? The brain is giving vague commands, and then please implement them, yeah? And so there is this concept, the concept of command neurons, that can represent a whole behavior. It's a neuron in the brain that when it's active, something happens in the animal. And then in this particular case, we have the animal crawling, and at some point the light will turn on. And as the light turns on, you will hear it's on, see what happens. It immediately starts crawling backwards. This neuron is on, it crawls backwards. This neuron consists of two pairs of neurons, actually, two on the left, two on the right. Of the whole brain, just two neurons are enough. What do these neurons do? They send their axons down, down, down. They descend into the spinal cord, right? The equivalent spinal cord. We call it nerve cord in the insects. What do they do there? It turns out it doesn't matter too much what they do there, because if you look at this neuron in the adult, which also exists, here is the brain of the adult fly and the nerve cord of the adult fly. This neuron will do exactly the same as you saw in the video from below, which I can play again. Here's a fly walking forward in this tiny little very constrained arena, so you can observe it well with videos. When the light turns on in a moment, it starts walking backwards. It's called the moonwalker. It's that very funny thing that Michael Jackson did. Yeah, I know, Drosophila also like music. And it's amazing because it's exactly the same neuron, but this is a limbless little maggot or a winged, limbed adult fly, right? And it doesn't matter. Fundamentally, that neuron embodies this concept of, I want you to go backwards. And whether your body has legs or not is an implementation detail. It doesn't matter, right? From the perspective of the brain, it's like, you do this, and you figure out how you're going to do it. Right. So how can we study now the brain? With electron microscopy. Something was pioneered here at the LMB for C. elegans, the nematode C. elegans, long, long time ago. We do it now in much bigger brains. What we're looking at here is simply fly through a chunk of the nerve cord of this larva that we're talking about. And as soon as it stops, I can show you what are we looking at. So each one of these little circles is essentially a cross-section on a neuron, on the arbor of a neuron. And then each one of these little black things here is a synapse. So here's another synapse, a bunch of vesicles that contain neurotransmitters, and they're going to be released. So here's the postsynaptic side, the dendrites, right? This is the axon. And then each one of these little circles is a mitochondria, the power cell of the cell. And then these long lines that you see in there are structural elements called microtubules. Think of them as the joists and girdles and walls of the cells so that neurons can keep their very fancy morphology. Right, so what we did is we acquired a complete volume of the whole central nervous system. So this entire little black thing here, measuring about 250 microns or so, or a bit less. And what you see now here is a fly through one cross-section, coronal section, and each one of these little whitey thingies is the soma that contains the genome of each nucleus. And then in there, in this mess of grayness, is where all the arbors come together and synapse onto each other. And so one needs to image the entire thing because you cannot just look at one neuron. You need to look at all the circuits, how neurons connect to each other. And you need to densely label all the neurons in the system and then be able to retrieve the circuits from that. So doing that 13 years ago, when this volume was acquired, was hard. Nowadays, here at the LMB, it can be done in a couple of months with a machine that is sitting downstairs, which is largely automated, and all it needs is a bit of babysitting, which is painful. I'm not going to downplay that, but it is 2 months as opposed to many, many months and years of work. Then how do we go about studying this nervous system? We start by, again, genetics. It's been the power of this model system for about a century. And then with genetics, we can label individual neurons. So here's one motor neuron, here's one stretch receptor. These are the ones that were used in that model from Jane Lovelace, right? And then we label them specifically because we have devised genetic driver lines that exactly do that. And then we find them in this cross-section of, in this case, the nerve cord. Again, remember, each one of these little white circles is the soma of a neuron, which measures about 4 or 5 micrometers. And so we find where they are, we reconstruct them. So here's the motor neuron with all its input synapses, here's the stretch receptor with all its output synapses. And the whole point of doing this exercise is not to have a high-resolution neuron, but to now, via the synaptic connections from one neuron to the next, reconstruct all the neurons that they are connected to. And of course, they connect to many things, but now you can apply graph theoretic techniques and figure out the so-called polysynaptic pathways, all the circuit pathways that go from one neuron to the next. And in this case, we start at the bottom from the stretch receptors, and then via multiple hops, we reach the motor neurons that are contracting the muscles and participating of locomotion in this animal. And then each one of these neurons here has a name. Neurons have names in this animal, like in the nematode, in C. elegans, because they are uniquely stereotyped and identified. From animal to animal, you find just that neuron. And because you have that uniquely identified and stereotyped neuron, you also have genetic driver lines that are expressed into just that neuron, which means you can now devise experiments to suppress the activity, to monitor the activity, to activate them at the wrong time to see what happens. Right? So you can now tinker with this nervous system to ask it questions. So we've done that for pretty much the whole CNS, mostly the brain. This is less dense than it looks in this beautiful rendering, but that connectome is now there and is useful for experiments. And instead of going into too much detail about what it does, I just want to tell you how it was done. Basically, we asked the whole community, dozens and dozens of labs, to please participate because it's so much work to do this by hand. And even though now there are, artificial intelligence approaches to reconstructing these neurons, they largely fail, and humans need to step in and do it carefully by hand. So we did about 40% of the whole thing. Many other labs did their big share of this. This means PhD students, postdocs, lab heads, technicians, everybody participated. And we did it in this platform. It's simply a Google Maps-like thing. You open your browser and you have your data there. You can navigate it, you can analyze it, you can trace the neurons and ask it questions. So with that, we mapped the whole brain. And once we mapped the whole brain, we realized we have a lot of neurons that descend to the nerve cord. Like that neuron, the moonwalker neuron, that was able to drive a particular behavior, right? So I started thinking about these descending neurons as the strings of the puppet. The brain is telling the nerve cord what to do. And before, we had this model in which, in neuroscience, you think of sensory neurons, and then there is a black box, magic happens, and output neurons. Output neurons, we'll get into details, meaning neurons that make something happen. And so you would have these two black boxes that are somehow related between the two brain hemispheres. You have left-right hemispheres in your brain. And then they will decide something and communicate it to the rest of the nervous system via these descending neurons. And if you look at it in detail, some descending neurons go to the so-called ring gland. This is the equivalent of the pituitary. It releases all these hormones that will change your metabolism, it will change your heart rate, it will change many things in your body via hormones. Then there is a subset that goes simply to what is called the upper part of your spinal cord, which essentially means you are controlling feeding behaviors, swallowing behaviors, and so on. And then you have the rest. And the rest, what are they for if not for locomotion? Well, most of them must be for locomotion because they go into the rest of the spinal cord to control your limbs, your body, your muscles, and your organs to some extent. And so now, because of that, it means we can predict what these neurons will do without even doing a single experiment. And I'm going to show you how we're doing this. So again, in behavior, suppose you're crawling forward. What do? You take your most posterior segment, your tail, and you contract it, and you propagate this wave of contraction forward, which means you need to trigger that contraction at the end, right? If you're going to go backwards, you do the opposite. You trigger the contraction in the anterior part of your body towards near the head, and then propagate that contraction backwards. If you want to turn, you need to stimulate a bunch of segments from the anterior part of your body only hemilaterally on one side, so you can actually, in a coordinated manner, turn your body. If you want to hunch your head, same thing, contract a few anteriors. If you want to modulate speed, you're going to have to change the biophysical properties of the neurons at every little segment so that when the contraction of the wave goes, it will go faster, it will go slower. And for head movement as well, it's like turning, but just limited to only the head. And so if you know that, you now have an expectation of, if I go back one slide, where these neurons should go. will actually tell you of what are they going to do. And so that's exactly what we did. And here it is, all these descending neurons across the whole nerve cord. And now what we have in this plot here in this blue, blue means how many synapses are these descending axons putting into that location of the body. And so you have a few neurons that put a huge amount of synapses at the very end. And maybe a few in the rest, but mostly at the very end. Well, these are the neurons that will either start or stop, forward motion. Again, the sign could be positive or negative, starting or stopping, we don't know. But then if you want to back up, you're going to have to do the opposite, put a lot of synapses into the anterior bits, these more thoracic anterior bits of the animal. If you want to turn, you'll have to have, again, a lot of synapses into a bunch of segments all the way to more or less halfway into the body, and so on, which means without doing a single experiment, just from observation, we can tell what these neurons should be doing. And then in addition, we can now go and look into the brain, one hop up, two hops up, meaning how many synapses you're jumping in the circuit layers, to try to see what is contributing to there. And this color-coded thing here doesn't matter, but it basically comes to say it comes from the learning and memory system, it comes from the innate responses system, it comes from olfaction, it comes from gustation. And we can now get an idea of who's controlling the strings of the puppet. And so, in summary, what we can say is that the brain is integrating all the senses, vision, olfaction, hearing, sense of touch, sense of temperature and whatnot, even sense of satiety. Are you hungry? Are you starving? Are you full? Are you thirsty? All of that is integrated in the brain and then collated into a network that will now choose to activate one descending neuron and suppress the others, because you can only do one thing at a time. I cannot go forward and backwards at the same time, right? And so that winner-take-all system will now activate some of these command neurons that will descend into the nerve cord, and they will go to one end or to the other end, or wherever they need to go, to then trigger particular central pattern generators, systems that are able to activate the muscles of this animal and make it move in the way that the brain has decided it should happen. So with that, I just want to finish here and acknowledge the Wellcome Trust, of course, the MRC LMB for all the funding for our research. And thank you very much. Looking forward to answering your questions.
Thank you, Albert. That was a really interesting talk. Has anybody got any questions? Or shall I? Oh, we've got a question at the back.
What you said was parts of the body can work with a brain. If you expand that to a human being, how many parts of our body do you think would work with their brain? Because it seems that they have to ask most things off. Is that because the body dies quickly, or does it just so they can get through it, or other parts of the body that could work if you could supply them with a signal, but they just don't because the brain's all missing?
That's right. So to repeat the question, if transfer these ideas into the human body, right? Basically, can the parts of the human body work independently of the brain, or would they be even alive without the brain? The one example I can bring right away is from people who unfortunately have had their spinal cord severed and they cannot walk. So nowadays it's not like the muscles are not there, or the nerves even are there as well. It's just there's no communication between the brain and those systems, right? So these command neurons have been interrupted. And so what some researchers have done is to provide sort of external electrical activation of such muscles and little computer programs that the person can activate either through their hands or even through some kind of implant directly to nerves that are still connected, and then they can walk. And this sounds like science fiction, but there are videos of this. You can look it up online, right? Which means if we understand the system, we can even provide such an aid for those who need it. A separate question is whether parts of the body can live without the brain. And here again, the unfortunate tragic circumstances where people are basically thought to be brain dead, and the body continues just fine as long as it has oxygen supply, enough energy like sugars, protein, etc. And so it's a matter of, can the body live without the brain? It can. The medical system has demonstrated that totally it can. The question is whether it's worth it, and that's for philosophy and ethics, to decide.
Thank you. Have we got any more questions?
Right, so interesting question about how does the octopus nervous system operate, right? We are actually studying squids in the lab as well, and there's a similar number of multiple limbs. What's common between, say, squids and octopus is every limb has its own little mini brain, a ganglion of neurons, an accumulation of neurons. And if this is severed, it will continue to operate that limb in various interesting ways, all independently, right? Frankly, I don't think neuroscience knows too much about how this works, but what we can hypothesize is that sort of the central pattern generators reside in those ganglions to operate the limbs, and then the coordination across limbs then is left into either interconnections between these ganglions or the manipulation of these interconnections from the brain. Again, giving a sense of direction, giving a sense of purpose to those motions. Developmentally, and genetically, they are 500 million years apart from us. Therefore, all bets are up into how exactly this works. And there's quite a bit of research on it right now. It's a very interesting field to follow.
Thank you. We've just got a question coming on the chat. So it says, hello. For the odor experiment in which the central nervous system neurons were inactive and the flies could not sense odors, would adding odor receptors in the downstream neurons whilst keeping the central nervous system neurons inactive, connected to the muscles, rescue the phenotype?
That's a very interesting question. And it's even more interesting because there are a number of sensory neurons that reach directly to the nerve cord, and without the brain, they participate and enable all these motions, including the stretch receptors, but also mechanoreceptors and temperature receptors. And so this animal will navigate a temperature gradient without a brain, for instance. So I don't know, the olfactory system is a bit different. It is difficult to sense the huge variety of possible odor and compounds, which is why you have a dedicated olfactory system that is able to disentangle these signals, right? Whereas, say, a temperature gradient is a much simpler stimulus to perceive and to measure. Same with light. This animal will sense light, intense, strong, kind of painful light. It will sense it as well through its skin, and therefore it will navigate away from it independently of the brain. But if you go to more complex stimuli like olfaction, I would assume it's not going to work, or at least it's not going to work well. It might be able to react, but it may not necessarily be very purposeful in how it reacts to olfactory stimuli.
Thank you. Any more questions? I think I had one quick question to finish, if that's okay. And just how this work that you're doing that's really interesting relates to human health and how it can kind of be transferred across? Sorry, that was quite a large question to finish on.
No, it's a very fair question. So it's a question that we have to answer when we request money from the taxpayer. How do we justify what we do, right? And what we're working out here are fundamental concepts, as well as about 60% of the Drosophila genome is shared with us. Literally 6 out of 10 genes we have as well. And so many of these genes have to do with how neurons are organized, how neurons connect to each other, how neural circuits are coordinated, how various sensations are integrated, and even the sensors themselves are homologous between a fly and us, despite hundreds of millions of years of difference, right? So ultimately what we provide is an understanding of these basic concepts from genes to neuroarchitectures, right, that enable us to then go to the human model, right, the humans us, and make sense of what we're looking at. Because at least at the moment, we're not able to map the human brain, not in any resolution that would be necessary to understand how it works, right? But what we can do is ask the subjects, make questions, and try to break that black box in other ways, in conceptual ways. And these conceptual ways are informed by all the experiments and results that we obtain by studying model systems.
Yeah, thank you. So I think that's all today. Thank you for coming. There's lunch provided afterwards, and Albert will be around to continue answering your questions. So thank you very much.
Thank you.